Assalamualaikum warahmatullahi wabarakatuh, and hi everyone. So today we are going to learn about the ionization of amino acids. So you have learned so far the characteristics of amino acids, and now we are going to see how amino acids are being ionized in a certain condition. So far, we have learned that that is the structure of amino acids with the alpha carbon, the alpha carboxyl group, the alpha amino group, hydrogen, and an R group. There are 20 amino acids with different R groups. So in this subchapter, we are going to see how in a certain condition, an amino acid can become ionized, which is with the carboxylate ion from the carboxyl group. So the alpha carboxyl group will become carboxylate ion. Okay, and another one is the alpha amino group that becomes the protonated amino acid with a positive charge. So altogether here, one negative charge and one positive charge. So one negative plus one positive, it will become 0. So when this happens, when one amino acid has one negative charge and one positive charge, it will have a 0 net charge. So it will become 0 net charge there. So when in this form, when it has 0 net charge, it is called as Zwitter ion. So we will see in the subchapter of this subchapter, we will see how the formation of Zwitter ion, which is the amino acid that has 0 net charge. Doesn't mean that it has no charge. It has charges, but with equal amount. For example, 3 negative charges and 3 positive charges that will become 0 net charge. So that is a Zwitter ion. Zwitter ion doesn't mean that the amino acid has no charge. Okay, so it just has a net charge, 0 net charge. And we will also see the titration curve of amino acid when this Zwitter ion is occurring. All right, so a little bit background about amino acids. So amino acids are amphoteric molecule in nature. So what does it mean? What does amphoteric molecule means? Amphoteric molecule means a molecule that can act both as an acid and also as a base. So since the amino acid can become negatively charged or positively charged or 0 net charge, so it can become both. Sometimes amino acids can become acid in a certain pH, and sometimes amino acid can also be base in a certain pH. So it depends. This acid and basic, it depends on the environment of the pH where the amino acid is in at that moment. Okay, for example, like this, it gives an example where the amino acid has 0 net charge at the pH of 7.4. So most of the amino acids will become a Zwitter ion, which is the 0 net charge in physiological pH. So physiological pH is commonly as 7.4 pH. Okay. So this is what happens. What happened when it becomes acid and base? So this is what happened. So in an acidic condition, which is a very, very low pH, okay, I'm just going to write it there, with a low pH, which is an acidic environment. So this is what happened. So in acid solution, you remember that they have a very high hydrogen ion concentration, right? A very high hydrogen concentration. So in that case, all of the, for example, carboxylate ion here, it will be protonated by the hydrogen to form a carboxyl group like this. It will become a carboxyl group. So from a negatively charged amino acid, for example, like this one, a negatively charged amino acid, with the protonation of the carboxylate ion, it will become only one positive. Okay, no more negative right here. Okay, because it has been protonated. So before this, before the association of hydrogen, it has one positive charge and one negative charge. So what is this? It has 0 net charge, which is a Zwitter ion, right? So with the association of hydrogen, which is the protonation of the carboxylate ion, it will be left with only one positive charge. So the net charge will be +1. So that amino acid will become a +1 net charge, no longer 0. So in this case, when it has a +1 charge, this amino acid will act as a base. Okay, when it has a +1 net charge, it will act as a base. So let us see if the amino acid was put in a basic solution. Okay, what will happen? Again, in basic solution, it has a very high hydroxyl ion instead of hydrogen ion. Okay, so this hydroxyl ion will be the protonated amino group here will be deprotonated to give the hydrogen to associate with the hydroxyl group to become a H2O. Okay, with that, this amino group will no longer be positively charged. So the net charge of the whole amino group, and the amino acid will become 1 negative, like that, because it is no longer positive here. So it will have a net charge of -1. So which in this case, this amino acid will act as an acid. Okay, so in conclusion, amino acids in acidic solution will act as a base, where in basic solution, the amino acids will act as an acid. So that is the nature of the amphoteric amino acid. Okay, so let us see this chart right here. Okay, so this is showing how an amino acid from an acid solution to become the zwitter ion and to become the amino acid in basic solution. So let's just look at it one by one. Okay, starting from the very low pH right here, right? Very low pH with high degree of hydrogen ion. Okay, so all of the amino acids, as we know, as I described at the above section, so all of the amino acids will be protonated because this is a very acidic solution, right? So the amino group will be protonated and also the carboxyl group will have the hydrogen there. Okay, so all in all, it has a +1 net charge. Okay, so imagine in a container or in a beaker, if you will, okay, you have 10 of the amino acids. For example, let's just make an example. Okay, so there are 10 amino acids in this container or a beaker. Okay, so at this point, all of the amino acids is 100% +1 charge. Okay, right here. So it has the highest concentration. Okay, protonated group is the most here, which is 100%. If there's 10 amino acids in there, so there's 10 of the protonated group right here. Okay, so again, at the lowest pH, it has 100% of all the amino acid has +1 charge. Okay, so let us see what happens if the pH starts to increase towards the basic pH. When it starts to increase, some of the amino acid here has started to deprotonate. It has removed the hydrogen because of the increasing of the pH. So when it removes the hydrogen, or another term is that deprotonated, so that amino acid will become zwitter ion. Why? Because the carboxyl group here has become carboxylate ion here with negative charge. So 1 negative and 1 positive, it will become 0 net charge, which is the zwitter ion form. So for example, right here, let me just change the color. For example, okay, out of the 10, 1 of the amino acid has started to become zwitter ion because it has been deprotonated. Why? Why does it? Why it has been deprotonated? Because the pH is started to increase, which means the concentration of hydroxyl ion is started to increase. Okay, so for example, like this. So if you see here, the zwitter ion has been increased here. Okay, because 1 has become zwitter ion, but 1 has become less of deprotonated. So it is decreasing here. So as the pH increases, more and more more and more amino acids is becoming zwitter ion, right? At a certain pH, half of the amino acid species has become zwitter ion. So this is happening here. Okay, so that pH here, okay, that is where this 50% of the species is zwitter ion, and another 50% of the species is still protonated group. So this is where it happens, okay, half and half. So that specific pH where this happens is called as pK1. Okay, let's just leave it there and then we will see the graph afterwards. So, but this is pK1. The pH, the specific pH where 50% of the amino acid has become zwitter ion and another 50% is still the protonated ion, the protonated amino acid. Okay, so in this case, this is where the amino acids act as a basic. Okay, because it's in pH, like I said, conclusion before, in acidic pH, the amino acids act as base. Okay, so half of them, 50% becomes zwitter ion, which is zero net charge. 50% is still basic amino acid. Okay, so this is where it happens here, right here. Okay, so as the pH keep on increasing, more and more of the basic amino acid has started to become zwitter ion. As as the pH increases, okay, pH increase, pH increase, pH increase until at one point, 100% of all the amino acid in that species has become zwitter ion. So this is where it happens. Okay, 100%, everybody has become zwitter ion, and zero of them is still a basic amino acid. None of them is basic anymore. So it ends just there. Okay. And then a certain range of pH, it remains as zwitter ion. Okay, the whole species, amino acid species remain as zwitter ion until the pH has reached a certain level of basicity. Okay, it has increased to a basic pH, and this is where some of the let me just change a quick change of color here. Okay, so some of the zwitter ion has started to further deprotonate. So what other can be deprotonated is the amino group here. So it will give out the hydrogen. So what will happen there? So the protonated amino acid, the amino group will become not positive charge anymore. It will be neutral like this. So when it is neutral, you have only one negative charge. So the net charge of the amino acid is -1, which is an acid. Okay, which is an acid. So as the pH increases, maybe it has started to become acidic. Okay, so this is where it happens. See, there's an increment of an acidic amino acid and the decrease of zwitter ion here. So as the pH increases, more and more of the amino acid will become acidic. So at one point, 50% of the species has become acidic amino acid, and 50% is still in zwitter ion form. So this is where this happened. Okay, 50%. 50% of them has become deprotonated acidic amino acid. 50% still remains zwitter ion as indicated blue circles right here. So those are all zwitter ions. Okay, so this is called as pK2. You will see pK1 and pK2 later on. There's a formula for it to calculate a pI. We will touch on pI later on. So as the pH further increases, more and more is being deprotonated. So last but not least, it will become 100% acidic amino acid, and none of them is left as zwitter ion anymore. Okay, so this is the kinetics of the amino acid from basic amino acid in pH, in acidic pH solution, all the way when pH increases, some of them is slowly becoming zwitter ion, and then as the pH further increases, some of it is slowly becoming acidic amino acid, and none of them has become zwitter ion anymore. Okay, so I hope you understand from this one. Okay, this is another just visualization. So this is in the acidic. This is an acid. Okay, in acidic solution, the amino acid is in cationic form, which is have positive charge, which act as a base. Okay, as it goes increasing in pH, it will become sweeter ion, which is have zero net charge. Okay, like that, one positive, one negative, and has zero net charge. And as the pH increases, it will have it will be further deprotonated, which will have a positive negative one net charge, which act as an acid, which is an anionic form, which is negatively charged. Okay, and then this we are the one that we have discussed before. We only address those amino acids with uncharged R side change. You have learned in the previous subchapter, which is the classification of amino acids. You can find that some of the amino acids out of the 20 amino acid actually has charged R group, right? There are polar and non-polar, acidic and basic amino acids. So all of those have charged and uncharged R groups. Okay, so for those that we are discussing now so far is the ones that has no charge ataupun uncharged R side chain. Okay, uncharged. But for those yang have charged R side chain, it has a different mechanism of how they form Zwitter ion and so forth. Okay, for example, this one, Zwitter ion happens here. For example, if that amino acid has charged R side chain, it has different range of pH where the Zwitter ion happens. Maybe it is shifted downwards, or maybe it is shifted outwards. So it has different pH range when it comes to amino acids that has charged R side chain. Okay, so far we are just discussing about those amino acids that has uncharged side chain. Okay, so we further look at the titration curve. For example, this one is an alanine, which is one of the amino acid discussed. So titration curve, this alanine is being treated with hydroxide ion. Okay, starting from the lowest pH with the lowest hydroxide ion here. So this is the concentration of hydroxide, and this is the pH. So the lowest hydroxide ion concentration is where the pH is the lowest, right? Kat sini, acidic. Okay, so we all know that in lowest pH, the amino acids are all in cationic form, which acts as basic amino acid here. Okay, as the researcher includes higher concentration of hydroxide ion, so the pH starts to increase, okay, to move towards the basic pH range. So as the hydroxide ion is added, all the positively charged amino acid is starting to be protonated here. Okay, like this one. Okay, it is sorry, it is being deprotonated here. Okay, so all these hydrogen will be deprotonated to become Zwitter ion. So in this area right here, okay, as the more hydroxide ion is being introduced, the higher the pH it goes, the higher the degree of deprotonation of the amino acids. So at that point, remember that I've mentioned before pK1, right? Okay, pK1, this is the pH where 50% of them are still in basic amino acid, and 50% of them has started to become a Zwitter ion right here. Okay, 50-50. And then as the pH increases, it will reach at a certain point where 100% of all the species has become Zwitter ion. As the pH further increases, more and more deprotonation happens, which is in pK2. Remember pK2, 50% are still Zwitter ion, but 50% has started to become acidic form of amino acid with -1 net charge. Okay, and also as the pH increases until the end, eventually 100% of the amino acid species has become 100% -1, which is acidic and in anionic form. So in this titration curve, we can see the progression of the pH of the amino acid from basic all the way to become acidic amino acids. Okay, through the protonation and deprotonation of the hydrogen ion there. Okay, so let me highlight one thing, which is the pi. So pi is called as isoelectric point. Okay, so that is the pi, isoelectric point. Isoelectric point is where 100% of the amino acid has zero net charge. So that if the pH, whenever this happen, we record the pH. So 6.02 is the pi of alanine. So that's how they identify the sweeter ion pH. The pH where alanine becomes sweeter ion, 100%. Okay, and pi can also be calculated as pK1 plus pK2. Okay, so this one, we take the pH, pK1 plus pK2, and then just simply divide by 2, you will get the pi, which is the pH in the middle. That is where all of the 100% of the amino acid becomes sweeter ion. Okay, so that is all from me for ionization of amino acid. I hope you had fun learning. Please go ahead and try out our assessments to test your understanding of this chapter. Thank you so much. Bye-bye.